So we talked about the physical examination of urine in Unit 1, which includes color, clarity, and specific gravity. And then we talked about the chemical examination of urine in Unit 2, which includes all of the tests on the reagent strips. So now in Unit 3, we are going to talk about the microscopic examination of urine, which is the analysis of the formed elements in the sediment of the urine. So formed elements would be cells, casts, so when I say cells, white cells and red cells, but also epithelial cells, bacteria, yeast, parasites, spermatozoa, crystals, and artifacts. And the microscopic exam is the least standardized and most time-consuming part of the urinalysis. So you'll have some subjectivity between techs, between MLTs maybe in how they categorize or quantitate certain elements. A lot of instruments now, automated urinalysis instruments, will have kind of a microscopic element to them, where as the instrument is processing the urine for the chemical results, it will also be taking images of the sample that you would view. So it takes those images, it kind of determines from those images what it analyzes and determines what elements are present in the urine, and then the tech comes along, views the images, and just kind of agrees or disagrees with that, with the instrument's interpretation of that. So there are some systems that can take some of that subjectivity out to a certain extent. Okay, so macroscopic screening criteria are going to be set up by the individual lab based on certain results obtained from the physical or chemical analysis to determine if a microscopic exam needs to be performed. So there are also certain patient populations that might warrant a microscopic, and that can be determined by the lab. So for example, a lab might determine every specimen from a pregnant female has to have a microscopic exam. So again, that kind of depends on the lab that you work in, the patient population that they typically see, and then the parameters that that lab has set up. Okay, specimen preparation. The microscopic should be done while the specimen is fresh because certain formed elements like your cells and casts can disintegrate quickly, especially if the urine is alkaline. Refrigeration can cause amorphous crystals that can obscure pathologic elements in the urine, so the urine should always be allowed to come to room temperature if it has been refrigerated. And then you should also make sure to mix the specimen well before analysis because of the cellular elements that could be present in the specimen that can sink to the bottom. And then you want to centrifuge the specimen once it's, and this is for the microscopic exam, once it's been centrifuged, all but a small portion should be poured off before you actually perform the microscopic examination. And there are commercial systems available, such as the one mentioned here called COVA, where they have specific tubes that are made for the urinalysis. So you centrifuge in these tube, and then when you pour off it, there's kind of like a little tip on the tube that holds on to the sample that you will actually use for the microscopic exam. And then there's also special slides that help control volume. So these slides are the size of a normal microscopic slide, but they're plastic, and they've got 10 chambers on this plastic slide. So you can perform 10 microscopic urine examinations on this, but it controls how much urine volume is in each square or in each chamber. So you can control that just a little bit more. Okay, specimen volume. The preferred volume for centrifugation is 10 to 15 mLs, and that's just in order to provide a representative sample of the elements that are present in the sample. So it says in your book that if any volume less than this is used, that there should be a note made on the report of the decreased volume, or that the results should be corrected before they're reported. Again, that's going to be something that your lab would decide, and criteria that they would set up, whether or not they're going to choose to put a comment on the report if a lesser volume is used. Okay, the speed of the centrifuge and the length of time the specimen is centrifuged should be consistent. 5 minutes at a relative centrifugal force, or RCF, of 400 produces an optimum amount of sediment with the least chance of damaging the elements. And it's not recommended to stop the centrifuge before it stops on its own, because it could cause disruption of the sediment before you decant the specimen. And usually labs are going to have special centrifuges that are made for centrifuging urines. So you can set or calibrate that centrifuge specifically for your urine samples. Okay, preparing the sediment. There should be between 1/2 and 1 ml of urine left in the tube after you've decanted the excess. The volume of the urine centrifuged divided by the sediment volume equals what is called the concentration factor. This relates to the probability of detecting elements present in low quantities, and it's used when quantitating the number of elements present per milliliter. Before looking at the sediment under the microscope, you should resuspend what is left in the tube after decantation by gently agitating the tube with a pipette. So once you centrifuge the sample, all of that, all the sediment's going to go to the bottom of the tube, and you're going to pour off the excess. So you're left with a good concentrated sample of that sediment. But since it's so tightly packed in the bottom of that tube, you want to just give it a gentle shake, or you can use a pipette to just gently resuspend. And this is an example of one of those COVA slides that I was talking about a couple slides ago, where it's plastic, it's the size of a regular microscopic slide, but you can see all of these little chambers here. So there's kind of like a little mouth at the front of each one of the chambers. So you would use a pipette to deposit the urine into that little mouth, and it just fills that entire chamber, and that's where you and you set that slide on the stage of the microscope like you would a normal microscopic slide, and then you just examine 1 urine per chamber there. So the volume of sediment to be examined, so whether you use a glass slide with a cover slip or a commercial system with the pre-made chambers, like the one pictured here, for examining urine, a consistent volume of urine should be used. So those commercially prepared chamber systems help with this, because as long as you make sure that the chamber is filled completely, the volume should always be the same. So that helps to provide some consistency. Examining the sediment, it's recommended that a minimum of 10 fields be examined on the 40x objective under low light when you're using bright field microscopy. And that's due to the fact that many of the elements that you would normally see in a urine are going to have the same refractive index as urine. So it makes it easier to see and identify those constituents if you're using a lower light. It's also helpful to try to find an epithelial cell to focus on, and that can help you make sure that you're in the right plane on the microscope. Almost every single sample that you view is going to have at least one squamous epithelial cell in it. So if you can find one to help you focus, then you can ensure that you're in the right plane. And the book says that it's important to view the urine under 10 and 40. I've never found this to be necessary. I don't know if other labs do that or not, because you're going to report the results from the 40x, but it may help you to initially get focused on the 10x if you need to do that. Okay, reporting from the microscopic examination. Terminology and methods of reporting are going to differ slightly among different labs. Some elements may be reported per low power field or high power field, depending on the facility. Some elements will be reported within a certain quantitative range, such as 0 to 5 or 5 to 10, and other elements are going to be reported in semi-quantitative terms, such as 1+, 2+, etc. But the idea is that you're looking through 10 fields and getting kind of an average, and that average is what you're reporting. Okay, correlating your results. So the microscopic results should correlate with the results of the physical and the chemical analysis. So if you notice a discrepancy between them, then you should investigate to see if there are any possible technical errors such as interfering substances or clerical errors. So for example, if you have a sample that is red and cloudy, and the chemical examination is negative for blood, or you're looking under the microscope and it's negative for blood, then I would look back to make sure, especially if you're processing multiple specimens at one time, make sure that you're looking at the correct results that match that sample. So you would expect the macroscopic exam, so the physical examination, to correlate with the results that you're getting for the chemical analysis, and for those to correlate with the microscopic examination. There is a table in your book that has a list of results that you should expect to correlate between the microscopic, physical, and chemical examinations. And then here's just kind of that list as well. So they're saying, how can you correlate color and clarity? Well, if the microscopic is showing blood and the chemical examination is positive for blood, then, and depending on how much blood is present, that sample is probably at least going to have a pink tinge to it, if not be red. Again, if you're seeing the reagent strip reaction for blood is positive, then you would expect to see red cells, possibly even red cell casts. If it's positive for protein, you might expect to see casts or some cells. Nitrites, you would expect to see bacteria, maybe white cells. So just being able to correlate those chemical results with what you would see on the microscopic examination. So this next little section of the lecture is going to be on sediment examination techniques. So some different types of microscopy, some different stains that can be used. So we're going to talk about that a little bit before we get into some of the actual elements that you would expect to see in a urine. So there are different factors that can affect the appearance of the urinary sediment under the microscope. Some of these things include cells and casts that are in varying stages of development or degradation. The chemical content of the urine can cause distortion of cells and crystals and artifacts. So because of these factors, sometimes the elements can be hard to identify. So the use of sediment stains or different types of microscopy can help in the identification of these elements. Because of the refractive index of elements in the urine is so similar to the urine itself, especially when you're using bright field microscopy, sediment stains can be used to increase the overall visibility of the elements by changing the refractive index of the elements. Stains can also be helpful in identifying characteristics of cellular structures such as nuclei, cytoplasm, and inclusions. So the first one that we're going to talk about is called Sternheimer-Malbin. It's the most common stain used in urinalysis. It consists of crystal violet and saffron and O. So this stain works by increasing the refractive index and making differentiation of the elements much easier. This stain is used to identify white cells, epithelial cells, and casts because the dye is absorbed so well by those elements. Toluidine blue is another one. This is a metachromatic stain, meaning that it will impart a different color on what it is staining than the color of the original dye. It is used to enhance nuclear detail, so it's helpful in differentiating between white cells and renal tubular epithelial cells, which originate from the renal tubules. We'll be going into more detail about those types of cells soon. This stain is also used to examine cells from other body fluids as well. Acetic acid can be added to the urine sediment. It will enhance nuclear detail, but it's also going to lyse red cells, so you would not want to use it during the initial sediment exam. but it'll help to distinguish red cells from white cells, yeast, oil droplets, and crystals. So, and if you're going to use this method, it's probably a good idea to pour over an aliquot and treat it. Okay, here's an example of a stain that has used Sternheimer Malbon. And this looks like it is probably a white cell cast, and then you can see what look like red cells in the background here. Here's a toluidine blue used on a skin biopsy. Okay, next, talking about some lipid stains, we've got oil red O and Sudan 3. With the use of these stains and polarizing microscopy, the presence of lipids, including free fat droplets and lipid-containing cells or casts, can be confirmed. Using these stains, triglycerides and neutral fats, which are free-floating fat droplets, they're going to stain an orange red, and cholesterol will not stain at all, but it will polarize. These lipids are usually going to be seen together in urine sediment, so a combination of the stain and polarization qualities can help confirm. Okay, gram stain is going to be used for differentiation of gram positive versus gram negative bacteria and to differentiate between bacterial casts versus granular casts. The Hansel stain is used to identify urinary eosinophils. Wright's stain can be used, but Hansel is preferred, which consists of methylene blue and eosin Y. Eosinophils in the urine are associated with drug-induced allergic reactions that produce inflammation in the renal interstitium. Staining is performed on a dried smear of a specimen that has been centrifuged or cytocentrifuged. And then finally, Prussian blue. This stain is used to confirm the presence of hemosiderin granules in the urine. And hemosiderin is an insoluble form of storage iron. It's a product of red cell hemolysis. These granules can be seen either in renal tubular epithelial cells or free-floating, and they normally are going to have a yellow-brown color. With the stain, they will appear blue. Okay, here's a picture of an oil red O, which again was one of the lipid stains. And this is from a liver. Sudan 3, which is also a lipid stain. So we can see an oval fat body here and a fatty cast. And this is helpful because you see a lot of red cells in the background here, so you might be inclined to think that this is a red cell cast, and there may be some red cells in there as well, but the stain is what helps you identify it as a fatty cast. And here's eosinophils stained with Hansel stain. Okay, talking about cytodiagnostic urine testing. So this would not be part of the routine urinalysis examination, but it can be useful for some different purposes. The slides, so you would prepare slides from a cytocentrifuged sediment, stain them with a Pap stain, and it can provide an additional means of detecting or monitoring renal disease. Can also provide definitive information about tubular changes associated with transplant rejection, information about viral, fungal, and parasitic infections, cellular inclusions, pathologic casts, and different inflammatory conditions. Most likely this is not going to be done by the techs in your lab. It would be done by like maybe a cytotechnologist or maybe even the pathologist. Okay, now we're going to talk about some different kinds of microscopy that can help with the urinalysis examination. So Brightfield is the most common type used for urinalysis, and it's really the most common type used in the clinical lab. Other types that can be useful for urine sediment examination include phase contrast, polarizing, dark field, fluorescence, and interference contrast. The type of microscope used depends on the type of specimen you're examining, the refractive index of the specimen, and the ability to magnify unstained living cells. There's a description in the book on the parts of the microscope. but we're not going to go into that now since we learned about that previously, some in microbiology, and you learned about it in Intro to MLT as well. But if you need a refresher, just read the section in the book. And then there's also this PowerPoint slide that shows you some of the parts of the microscope as well. So types of microscopy. So bright field, most commonly used microscope in the clinical lab, so not just in urinalysis. Objects appear dark on a light background, but this type can present problems if the light is not properly utilized. So as we said before, the refractive index of the constituents of the urine is very close to that of the urine itself. So elements can be missed if the light is too bright. So because of this, you want to make sure to use a decreased amount of light, and the light should be controlled with the rheostat, not by moving the condenser up and down. And of course, staining the urine sediment will also help with the visualization of these elements if you're using bright field, but not all labs are going to offer that option to perform these stains. So again, there's just that picture of the your basic bright field microscope and all the different parts. Here is a picture taken under a bright field microscope of a hyaline cast, and you can see how, especially hyaline casts, they can really blend into the background sometimes. You can see kind of the outline of the cast there, which is referred to as the cast matrix. So you can make that out. Sometimes, though, that's very difficult. You can also see a squamous epithelial. Here looks like there's some cellular elements in the background. It gets kind of fuzzy. It also looks like there's probably mucus, but a lot of those elements can be difficult to see under bright field unless you're using lower light. Okay, phase contrast. This type of microscopy increases the refractive index by increasing the contrast, and that's done by using a bright field microscope with a phase contrast objective lens and a matching condenser. So this type of microscopy is especially helpful for identifying low refractive hyaline cast or mixed cellular cast and mucus threads. With polarizing microscopy, this can be useful in the identification of crystals and lipids. You've got a polarized light beam that is used to produce characteristic colors in crystals and form a Maltese cross pattern in lipids. These elements seen under polarized light are known as birefringent, which means that as the light passes through these substances, they are capable of splitting the light into two beams, with one beam at a 90-degree angle to the other one. Polarized light is obtained by using polarizing filters and bright field microscopes that can be adapted to polarize also. Okay, so here's a picture of, or an example of phase contrast. So you can see how much stronger that contrast is between the element and the background. Here's an example of polarizing microscopy that exhibits that Maltese cross. So that's what that little formation there is called. And polarizing microscopy, an image of uric acid crystals. Okay, next is interference contrast, which provides a 3D image that has very fine structural detail. It does this by splitting the light ray so that the beams pass through the different areas of the specimen. The object is going to appear dark against a light background. This method is not routinely used because it requires extensive modifications to bright field. Then you've got dark field, which is used to enhance visualization of specimens that are not easily seen with bright field. So it can be used for unstained specimens and especially helpful in identifying Treponema pallidum, which is the spirochete that causes syphilis, penta, and yaws, where the specimen is going to appear light against a dark background. And then finally, fluorescence microscopy, which is used to detect bacteria and viruses within cells and tissues. The objects will appear bright against a light background with high contrast when a UV light is used. These scopes require very powerful light sources, so again, you're probably not going to encounter them very often, if at all. Here's a picture of interference contrast and you've got epithelial cells. So again, just very high contrast there. It really makes the image stand out. Dark field with showing a red cell cast. And then fluorescence showing our little buddy E. coli. Okay, I'm going to stop here for this lecture. The next lecture we'll pick up where we're talking about the actual urine sediment constituents.